Hello, my name is Beatrice Liwanag. I'm currently a junior at Schrier School. I love studying biology, and it's always been my dream to go into the medical field and become a pediatric cardiologist. For this experiment, over the course of about 10 days, I performed a bacterial transformation lab with the help of my awesome AP biology teacher, Mr. Tangway. This is a classic yet extremely informative lab that demonstrates the benefits of genetic engineering on society. Through lateral gene transfer, bacteria can share and pick up genes when they are under stress. They pick up plasmids, which are small circular DNA fragments. Molecular biologists made the discovery that they could design plasmids to influence bacteria's gene expression, which is what we did for this experiment. We used a plasmid that contains a gene to present green fluorescent protein, and we heat shocked it into our bacteria, E. coli. We had multiple selective media in order for the bacteria to perform the function that we desired. For instance, we mixed ampicillin, which is an antibiotic, into the nutrient agar plates, where the bacteria would grow. This would ensure that the bacteria that took up the plasmid would live, since the plasmid has a gene that is resistant to ampicillin. We also mixed arabinose into 2 plates. Arabinose is a sugar that will act to promote gene expression, specifically of green fluorescent protein, which is in the plasmid. We had to set up controls so that if the results weren't what we expected, we would be able to figure out which factor contributed to that. We had plates with LB nutrient, plates with LB nutrient and ampicillin, and LB ampicillin and arabinose plates. Once the bacteria was grown, we mixed 4 colonies with the plasmid into ampicillin and ampicillin arabinose plates. We had 1 ampicillin plate that didn't contain the plasmid, and the expected outcome was that the bacteria would die in this environment. However, during our first run of this experiment, this did not happen. The bacteria ended up growing, which meant that the ampicillin was not functioning in the way that it was supposed to. The second run of this experiment proved to be successful since we used a new stock of ampicillin, and we had our glowing bacteria. And you can see the little alien with the pageant crown right here. This process of genetic engineering proved to be significant for medical developments and access to treatments. For example, insulin used to be retrieved from the pancreases of cows and pigs. This was not only expensive, but dangerous since people tended to have allergic reactions to it. With the help of genetic engineering, scientists can use transformation in common bacteria like E. coli to increase production and availability to patients who need medical treatments like insulin. Thank you.